Good morning, everyone, and welcome to today's GCSE biology session on cells and organisation. So it's just gone 10 o'clock, so I think we will all get started. So thank you all for joining me. I know it's very early on a Saturday morning. This is our first session of the term. So I hope you guys are all settled back into school and I hope you guys are enjoying it. So I'm going to be teaching today's session and my name's Roshni. And if you've not met me before, just a little bit about myself. I'm a doctor currently working in London, but I've also been a tutor for many years now and I love teaching science. So we're going to kick off today's topic. As I said, it's on cells and organisation. So this is just a brief timetable for those of you who haven't joined our classes before. So the session starts at 10 and we will finish at 2. There is a half an hour lunch break from 12 to 12:30, so do keep an eye out for that. And there are short breaks during the course of the session as well. Okay, so how is this going to work? So guys, this is a very live and interactive session. This is not just like, watching a YouTube video or, taking a back seat. I want you guys to really participate. So every time you guys see this little man, that is a question for you guys, and I want you to put the answers in the chat. We will be doing lots of exam practice, so you guys should have received a question pack before the session, and we will be picking out a mix of questions, some easy ones and some hard ones as well. I know we don't have our exams coming up soon, but it's just good to get an idea so that when the exams come around, your technique is much improved. And my colleague, who's also a doctor, is also sitting in on today's session. So if you have any questions, please just ask Ace. I don't want anyone to be confused or to just be sitting there not understanding. So please just ask. And you will get the recording afterwards as well. So what I want you guys to do is just grab a scrap paper and a pen. You don't need to be scribbling everything down and making loads of notes, but there will be a few things that I will be asking you to draw or to write out, so it can be handy just to have a pen and paper. Okay, so what are we doing today? So guys, I know it's a very early start to the term. We will be covering all of the topics from the GCSE curriculum, but the first topic, which I think is not too bad, is cells and organisation. So hopefully you guys may have done a little bit of this before. We'll be talking about cell structure. We'll be talking about eukaryotic and prokaryotic cells. We'll then be talking about animal and plant cells and microscopy and magnification. And then in the afternoon, we'll be doing cell division, which is a little bit more harder. So we'll be talking about chromosomes and mitosis and stem cells and cell differentiation. So it will get harder as the day progresses. Hopefully this stuff is a little bit easier for you guys, and this is where it will get challenging, but we will go through it from the basics, and by the end I hope we will all be experts. So okay guys, so before we get started, I just want to see, where are we all at? How much do you guys know about the topic cells? So hopefully you should be able to see a poll coming up on your screen. Okay, and I'll stop you guys there. So most of you have said C. I know most of it, with a few of us saying a little bit, and some of you very confident ones saying that you know it all. Okay, so I will be testing you guys. So let's get started. Okay, so a nice and easy, straightforward question, just to check we're all awake. What is a cell? So I want you guys to put your answers in the chat, and I will pick out the best ones. So when you hear the word cell, what do you think of? Okay, I'm getting some really good answers. So cells make up the body. Yep, that's fantastic. Fantastic. So basic unit. They make up living things. Fantastic. They have different structures. Super. And they've got different functions. And that's right. So cells are the smallest living unit. Okay, so they are basically the building blocks of life, and they are the smallest basic unit that we have in our bodies, and not just in animals, but also in plants. Okay, fantastic. So just another picture. So what do you guys think all of these have in common? So I've got 6 pictures on here. What do we think they all have in common? Fantastic. So lots of replies. So exactly. So all of these organisms here, we've got some animals and we've got some plants as well. All of these are made up of cells. Okay, so cells are the simplest living unit, and this is how we differentiate living organisms from other objects. So you'll know things like your laptop or the pen that I'm holding, they are not made up of cells. Cells are only occurring in living organisms. Okay, and they are the basic building blocks. Okay, so when we look at cells, we've got animal and plant cells. Okay, now all animal and plant cells are eukaryotic. Okay, so does anyone know what do we mean by the term eukaryotic? Okay, so if you've never heard of this term before, don't worry. Okay, so when we talk about cells, okay, we said that cells are the basic unit of all organisms, but when we have cells, we can broadly divide them up into 2 types. Okay, and we will talk about this. So cells can be divided up into eukaryotic, okay, and something called prokaryotic. These are our 2 types of cells. Now, eukaryotic cells means that they have a nucleus, okay, and prokaryotic cells do not have a nucleus. So within eukaryotes, we have got animal cells and we have got plant cells, okay, because both animal and plant cells, they have got nucleuses, okay, and we will talk about this. So eukaryotic cells, as I said, they describe cells that contain a nucleus. And therefore we say that a eukaryote is an organism that is made of eukaryotic cells. So for example, if we take our own human bodies, we're all made up of cells, and all the cells that we have contain nuclei. Fantastic. So that is the main difference between eukaryotic and prokaryotic, and you guys have said some really good examples as well. So just for now, I want you guys to remember eukaryotes are in animals and in plants. Okay, so there are prokaryotic cells and they don't have a nucleus. Does anyone know what kind of organisms are prokaryotic? If animal and plants are all eukaryotes, what could not have a nucleus? Super, fantastic, guys. So all bacterial cells are prokaryotic, so they do not have a nucleus. Okay, and we're just going to talk a little bit about this diagram over here. So this is a bacterial cell. So this one over here is the 3D version. Again, if you were to just take a layer of it, like a cross section, it would look like this. Okay, so this is the 2D version. Now there are some things on here which I want us to go through. Now, whenever you look at a cell, classically, you might have seen, your animal cell that looks like this. The bacterial cell looks much different. So can anyone tell me what things are in this bacterial cell that wouldn't be in an animal cell? What can you notice looking at the labels? Excellent. So what we can see here is that we've got one layer around it. Okay, and then we've got another layer. So very interestingly, they have got a cell wall. Okay, that's the first thing. So they've got a membrane and then they've got a cell wall. Now, usually in all animal cells, the DNA sits inside the nucleus, the genetic information. Okay, sits inside the nucleus. What we can see here is that there is this DNA that's just chilling in the middle. Okay, it's just sitting on its own. It's not wrapped in any nucleus. It's a single DNA loop and it's free in the cytoplasm. Okay, so the DNA is free and it's not sitting in a nucleus. We've got some other bits. We've got these blue dots, which are ribosomes. Does anyone know what a ribosome does? Excellent. So what ribosomes do is they are important for protein synthesis. Okay, they're important for making proteins. Fantastic. Okay, so we'll talk a bit more about the structure of bacteria, but what I want you guys to remember is, as I said, eukaryotic cells are in animal and plants, and prokaryotic are bacterial cells. Okay, that is the main significant difference. So a few key points about prokaryotes. So firstly, they are single cells. So when you think about your bacteria, okay, so lots of infections that occur in our bodies, they are caused by bacteria. So for example, whenever you get a cough, that could be caused by bacteria. Now these bacteria, they're not made up of lots of different cells. When you think about your human body, we've got millions of cells, haven't we, to make us work. When you think of a bacteria, it's just one cell. Okay, so we say it's single-celled. And what that means is that bacteria are obviously very, very small and you can't see them with the naked eye. So what do you think we need to see a bacteria? And we will be talking a bit more about this, but what can we use? If we can't see it with our eyes, what can we use to see a bacteria? Fantastic. So we need microscopes, and well done to those of you who are naming the different types of microscopes. We will talk about them. Okay, so guys, there are 4 things about prokaryotes which we need to remember. Okay, and I'm going to be quizzing you guys on this at the end. Okay, so we need to remember them. The first one is that they are single-celled. The second thing is that they do not have a nucleus. Okay, as I said, the DNA is just chilling and it's just free. Okay, there is no box that is enclosing it. It sits on its own. They're also much smaller than eukaryotic cells, and that is very obvious. So cells that are in our bodies, like in our blood cells or in our muscle, they are much bigger. And there's one last thing about prokaryotes. So what you can see on this diagram, which I haven't yet touched on, is that there are these rings. Okay, and usually there's only one, or sometimes there's usually only one in each cell, and this is known as a plasmid. Okay, does anyone know what the point of plasmid is? What does the plasmid do? Excellent. So the plasmid is a ring of DNA. That's right. But why is there a ring of DNA, and why is there also a loop of DNA? What is the point of this extra DNA? Okay, so this is a bit of a challenge question, but basically what happens is if we've got one bacterial cell, so if you imagine you have got a cough, okay, because you've got a chest infection from a bacteria. Now that bacteria comes into your body. Okay, this is the plasmid and this is the free loop of DNA. Now what that bacteria wants to do is if it really wants to infect you and make you sick, what does it need to do? It can't, it's only one right now, so what does it need to do? Exactly, it needs to reproduce, right? You want to make lots more bacteria so that it can make you sick. That's what it thinks of doing. So what it does is this plasmid, it contains the DNA, but this plasmid can actually move from different bacteria. So another bacteria that's over here, okay, this DNA can be transferred onto another cell and it will allow it to replicate. Okay, so they can easily transfer and they can easily make lots of bacteria. And before you know it, you can become very sick. So that's how bacteria work, and that is because of the plasmids. Okay, so guys, just a quick recap on our prokaryotes. So in our prokaryotic cells, we have got 6 parts, okay? So what I'm gonna do is just quickly draw this out for you guys. Okay, and do draw this along with me, guys. I think it's always really helpful. The more diagrams you draw, the more it can stay in your head. Okay, and this is my plasmid over here. And we've got a few other dots just chilling. Okay, guys, so let's start off. So we have got cytoplasm. What is the point of cytoplasm in my cell? What does cytoplasm do? So cytoplasm, we haven't actually talked about it, but cytoplasm is actually all of this. Okay, so inside the cell, there is a whole area of cytoplasm, and this is basically a fluid-like substance. Okay, it's literally liquid. Okay, so if you think about it, when you go for a bath, when you sit inside the bath, you're surrounded by lots of water, right? And that is basically what the cytoplasm is. Okay, so it's just like the cell is just being bathed in all of this liquid, and this liquid is really important. So this fluid is part of the cell inside the membrane, and most of the chemical reactions in this cell happen. So can anyone tell me what is a chemical reaction that may occur in a cell? If you can name it, I'd be really impressed. So some of you have said mitosis. So mitosis is not a chemical reaction. It is a form of cell division, but it doesn't actually take in the cytoplasm, but good try. Any other answers? So I think we're getting there. Fantastic. So respiration, we will talk about, takes place inside the cytoplasm. Okay, and we've got another answer which says photosynthesis. So that is only in plant cells, guys. So we will come on to that. Okay, fantastic. So respiration takes place inside the cytoplasm. Okay, so next up is my cell wall. What is the point of my cell wall over here? Why do you think we would need a wall to the cell? Fantastic, it gives extra support. Okay, so the cell wall in plants is actually made of something called cellulose, but the cell wall here is not made of cellulose, but it is very strong and it's just there to give it support. Because remember what we said is that bacterial cells are single celled, so they're on their own and they're only made of one. So it's really important that they've got a good protection layer to protect them from the outside. Okay, we've then got our single DNA loop over here. Okay, why do we have DNA in cells? Fantastic. So the single DNA loop is that which carries the genetic information so that the bacteria can divide. And then we talked about our plasmids. So these are small circular sections of DNA and they provide genetic variation. So it allows the bacteria to pass on some information to another one and it allows it to reproduce. Okay, we've then got our cell membrane. So the cell membrane controls what substances go in and out of the cell, and it also has internal extensions that have enzymes attached to them, and we'll talk about the role of enzymes. Okay, and finally we've got our ribosomes, and as we've already said, ribosomes are where proteins are made. Okay, and proteins are really very important for the structure of the cell, but also for lots of processes to take place. Okay, guys, and that is our prokaryotic cells in a nutshell. Does anyone have any questions on this? Okay, so let us move on to animal cells. Okay, so guys, we've talked about prokaryotic. We're going to talk about eukaryotic cells now. So as I said, in terms of our eukaryotes, we've got plant cells and we've got animal cells. Okay, so they can be single-celled, i.e. unicellular, so they're on their own, or they can belong to multicellular organisms. So obviously things like us humans or other animals and plants as well. Okay, so lots of organisms have got eukaryotic cells. Now, the main thing that defines a eukaryotic cell is that they have a nucleus and they are much larger. Okay, so guys, there were 4 things that I said we knew about prokaryotic cells. So can we just recap them, guys? So I want all of the 4 points that we said so that nobody gets mixed up between eukaryotic and prokaryotic. Okay, so we had our prokaryotes over here, and remember there were 4 points. So what did we say? Excellent, they're single-celled. That was the first one. What else can you guys remember about that bacteria? Yep, they are smaller than eukaryotes. Fantastic. No nucleus. Super. And they've got plasmids. Well done, guys. Okay, I'm really impressed. I know it's a Saturday morning, so well done. Those are our 4 points about prokaryotes. We are moving on. Okay, we're now talking about eukaryotes. And these are in our plant and our animal cells. So these are usually multicellular. Okay, so always think about you guys. If you look at your hands, it's made up of skin, of muscle, of nails. So many, many cells. Okay, they are much larger. And they've got a nucleus as well. They don't contain plasmids typically. Okay, and this is our cell here. Okay, so this is our typical animal cell, very basic. Okay, so you guys can just quickly scribble this down on your scrap piece of paper, and we will then label this together. So firstly, in my animal cell, what is this on the outside? Okay, nice and easy. This is my cell membrane. Okay, and then what is sitting here in the center? Exactly, my nucleus, which contains the DNA. Okay, and the entire fluid bit, so all of this light blue bit, what is all of this? Super, so this is known as my cytoplasm, okay, where the chemical reactions take place. And then I've got these pink bits. Can anyone guess what these are? Fantastic. These are my mitochondria. Okay. There are also which they haven't labeled on this diagram, but they're also, we can just pretend that they are green. They're also ribosomes as well, okay, for proteins, because obviously we need proteins. Most of our body is made up of proteins, like our muscles. So we have got ribosomes as well. Now, a few of you guys are saying chloroplasts. So guys, chloroplasts are not in the animal cell, okay? They're in the plant cell, and we will talk about the plant cell next. Okay, fantastic. So these are the 5 bits of my animal cell. And yeah, they haven't labeled the ribosomes on here. Okay, but we've got my cytoplasm, my nucleus, my cell membrane, and one mitochondria is called a mitochondrion. And if we've got loads, we call it mitochondria. And this diagram here is very, very nice, much better than my drawing. And you can see it much more clearly on here. So these wavy bits are the mitochondria. Okay, and these green blobs are the ribosomes. So this is a very nice diagram. All right, guys, so just a quick question for you guys, just to make sure we're all nice and awake. What is the point of the cell membrane? What does it do? Fantastic. So pretty much everyone has got that question right. So the cell membrane controls the movement of substances in and out of the cell. Okay, so guys, I think you are all finding this very easy. So I'm glad, but it is going to get harder. So well done. We're just consolidating the basics for now. Okay, so that is the correct answer. So this is my cell membrane here, just around the outside. Okay, now the cell membrane basically is controlling what is going in and what is going out. So what kind of things do you think need to go inside your cell? Okay, let's just imagine this is a cell in your muscles, okay, in your biceps. So what needs to go inside this cell? Does it even need anything, or is it happy on its own? Okay, what kind of things go inside a cell? Okay, so some of us are saying starch. So starch is something that we would actually consume. So not specifically starch. There's only very small things that can go in and out of a cell. So you're nearly there. Okay, so some of us are saying enzymes. So guys, enzymes, they stay inside the cell. They don't go popping in and out. What kind of things do you think we need from the outside to go inside our cells? Fantastic. So glucose, okay, glucose is our sugar, okay? So if you want this muscle to move, okay, it needs energy, right? So sugars can go inside the cell. Fantastic. Water. So water can go inside the cell. It can also come out. Water can go in and out of the cell. Excellent. Things that can come out are other gases. So things that might come out are carbon dioxide. Okay, we will talk about why carbon dioxide comes out of the cell. And also ions go in and out. So things like sodium, potassium, okay, and lots of different ions go in and out. So ions are really, really important for all of our cells to make sure that the cells can behave in the right way. And you'll know that if, for example, you're really, really dehydrated, you may be encouraged to, like, if you've done like lots of exercise, you may be encouraged to drink Lucozade or something like that, because there are lots of ions in there which help your cells go back to their normal function. Okay, so ions are really important. So the cell membrane is basically controlling what is going in and out, depending on what the cell needs and what it wants to get rid of. Okay, so things like glucose, and oxygen is the main one. Oxygen that we breathe in is going inside the cell, and other bits are coming out. Okay. Secondly, in our animal cell, we have got our cytoplasm. So we've talked about this already. Cytoplasm is basically like a jelly-like substance, and it is where there are lots of nutrients for the cell, and it's where a lot of the chemical reactions in the cell will take place. Okay. And then our nucleus. So guys, as we know, in our animal cell, what is the point of the nucleus? Fantastic. So your nucleus contains the DNA, controls the activities of the cell, controls the function. Fantastic. So really good answers, guys. Well done. So I like to think of the nucleus as basically the boss, sort of like your head teacher, that basically controls everything and tells everyone what to do. Okay, so controls all the activities of the cell, contains the cell's DNA. Okay, so for bonus points, guys, and we'll talk about this in cell division, what does DNA stand for? Does anyone know what DNA stands for? And if you can spell it right, I'll be really, really impressed. Okay, the A stands for acid. Well done. Okay, so you get half a point for that. Okay, some of you, the spelling is a little bit off, but well done. So it's deoxyribonucleic acid. Okay, so it's a bit of a mouthful. Well done. So that is what our DNA is, and it looks like these strands. Okay, so the nucleus is found in the cytoplasm, so it's just chilling in there, but it's got its own membrane. Okay, it's got a nuclear membrane, and inside is the DNA. Why do you think that the DNA needs to sit inside? Why can the DNA just not lie free like in the bacteria? Why do you think it needs its own membrane? Excellent. For protection, for safety, it may react with cytoplasm. Excellent. So the DNA is very delicate. Okay, if you imagine, guys, without our DNA, we wouldn't be able to grow, we wouldn't be able to reproduce. So it's really important that it's looked after and it's protected. So this just provides, just like the cell membrane, it just acts as a barrier so that all of the fluid and all of the reactions in the cytoplasm don't get mixed up with the DNA. Okay, fantastic. And its DNA is arranged in chromosomes. Okay, we'll talk about cell division when things get a little bit more complicated. Okay, and then finally the mitochondria. Okay, now the mitochondria are important for what, guys? So the answer is on here, so I'm going to go back. Why do we need mitochondria? What process happens? To provide energy. Fantastic. So if we go back to our muscle, so if you think about your muscle, if your muscle wants to move, it's not going to move on its own, right? It needs energy. And the only way that the cell can release energy is with our mitochondria. Okay. Now, mitochondria are what we call organelles. So organelles are basically the parts inside the cell. So all of these are organelles, and they are only present in eukaryotic cells. Okay, so when you think back to that bacteria, there was no mitochondria, right? It was just cytoplasm. This is only in eukaryotes. And basically, respiration takes place here. So when you think of mitochondria, just think of a battery. Okay, it's providing the source of energy. And if you don't have mitochondria, you will the cell can basically die. So does anyone know what kind of cells need lots more mitochondria? And I've already given one answer. So what cells need lots and lots of energy? Fantastic. So things like muscle cells and sperm cells. Okay, why? Because muscle cells, obviously, we are moving all the time, so we need more energy. If you imagine like the cells in, for example, your hair or in your nails or in your skin, they don't need that much energy because they're not doing anything, but your muscle cells are moving, so they need energy. Similarly, the sperm cells are moving as well, so they need lots of mitochondria. Okay, and somebody has said that ribosomes have got lots of mitochondria. So guys, ribosomes are a part of the cell. Okay, they're not a type of cell, they are an organelle inside the cell. Okay, and just one last thing I want to explain to you guys is that mitochondria, if you look at it, this is actually what a mitochondria looks like. Okay, so I'm just going to draw my own bigger version. It's like this, and it's got this squiggly line inside. Does anyone know what the point of these squiggly lines are? Why does it have those folds inside of them? Why can't the mitochondria just be like little blobs like this? Why would they make these lines in between? So even if you don't know, this is a challenge question. This is like a level 9 question. So if you've got no idea, just take a guess. Okay, I'm super impressed. So one person has managed to get this. So guys, basically, if you think about it, the mitochondria, they want to make energy, right? Now, they want to make as much energy as possible. And so to try and maximize that and to really get out all the energy, they have these thing called folds. Okay, and these folds, they increase the surface area through which the reactions can take place. So instead of just being like a flat blob like this, they're stretched out and they've got these squiggles so that the reactions can take place on lots of different areas of it. Okay, so mitochondria can release more energy. Okay, does anyone have any questions so far? Okay, so just before we go for a break, guys, so we need energy for lots of different processes. Okay, so I've already given you one that is for movement. Why else as a human do we need energy? What else can you guys think of? Or even in plants or in any organisms, what is the point of energy release? Fantastic. So we need it to function, but what else? What to do activities? Yeah, so sperm cells need energy. Fantastic. To exercise. Yeah, so we've talked about muscles to move. What other things do our bodies do? Fantastic. So there are a few different things. So firstly is cell division. Okay, if for example you cut yourself and then you will notice that you get a scab straight away, right? And the skin grows back. So that is because we use energy for our cells to divide and so that we can grow. Okay, that's how humans grow and get bigger through cell division. And we need energy to do that. Okay, for muscle contraction, for movement, and also for growth in plant cells. Okay, so energy is needed for loads of different processes, and the only way we can release it is through the mitochondria. Okay, and we've touched on this, but what is the name of the process that releases energy from in the cell? So what is the name of the process that's actually happening in the mitochondria? Fantastic. So the process is known as respiration, and most of the time it is aerobic respiration. Okay, and aerobic, I like to keep things simple. So aerobic simply means it uses air, i.e. it uses oxygen. Okay, so essentially what is happening is each time, okay, let's just take our basic cell, our nucleus, our ribosomes, and then we have got our mitochondria. Okay, they're a little bit big. But these are mitochondria. Now, what is actually happening in each of these mitochondria is the same process, just happening a million times. So essentially what happens is we get glucose. This is a sugar, okay? So you can get sugars from a sugary drink, from cakes, biscuits, any food that we eat, we take it in, okay? So your sugar goes in. Glucose is going in. You then take a deep breath in, and that is how oxygen gets into your body. Okay, so remember we said that the cell membrane allows things to go in and out. Oxygen goes in. And now the mitochondria are very, very clever. So they mix these two things, the glucose and the oxygen, and then three things come out. Does anyone know what comes out when they mix the glucose and the oxygen in the mitochondria? Okay, so I'm going to give you guys a clue. One of them is a gas and one of them is a liquid. So very interestingly, what happens is that the sugars combine with the oxygen and they give off a very, very strong reaction. They give off carbon dioxide gas and they give off water. Okay, so you might think, well, what's the point? There's some gas being given off and some water, but the other main thing that they give off is energy. Okay, so this reaction is a very, very strong reaction and it releases a lot of energy, and this is what we want. So energy gets released in these mitochondria and it's what we use. These two are what we know as waste products because we don't need the carbon dioxide. So carbon dioxide comes out. Okay, and water also comes out. Okay, so these two things go in. These are my reactants. Okay, and these are my products. This is really the basics of cell biology, and if there's anything that you remember from the end of today's session, it should be this. Okay, I'll be really happy if you can remember that. So we've got glucose plus oxygen gives us carbon dioxide, water, and then we get our energy. Okay, we use this energy for processes, as I said, cell division, muscle contraction, and growth. Okay, really, really common 2-mark question. Okay, guys, so just a challenge question. Do we know what is the chemical equation for aerobic respiration? So a little bit of chemistry coming in here, but I want you guys to give me the symbol equation. Does anyone know how do we write glucose out? Okay, really good answers. Okay, I'm super happy with this. So glucose, you might just think it's a simple sugar, but it's a bit complicated. It's C6H12O6. Okay, it's made up of carbon, hydrogen, and oxygen. And normally we draw glucose as these little boxes. Okay, oxygen, guys, tell me, what is my formula for oxygen? Hopefully we all know that one. And you can see it over here. Fantastic. Oxygen is O2. CO2, you can see, is CO2 here. And water is H2O. Okay, now how do we balance this? What you can see is I've got 6 carbons here. So I just remember this as the rule of 6s. Okay, so basically all you have to do is put a 6 here, a 6 here, and a 6 here. And this one doesn't have a 6 because it's already got 6s at the bottom. That's how I remember it. So that is the chemical equation, and well done. Most of you guys have said that, so I'm really happy with that one. Okay, so let us just look at an exam question. So as you guys can see, this is really, really always coming up in the exams. So plants need chemical energy for respiration and for active transport. Write a balanced chemical equation which represents the process of respiration in plants. Okay, and this is 2 marks. Okay, so I want you guys to all do that. I'm not going to move on until everyone has submitted an answer because I've just shown this to you guys, so I don't want anyone to get this one wrong. And just have a go. And with these exam questions, they can kind of sometimes throw in random things like, oh, which is the one in plants for active transport? So don't get muddled up. Just think of your basics. What is happening in respiration? Okay, just think of your basics, because sometimes in exams, even if you've studied the material, I always get a bit confused when I used to do the papers, but just think about the basics. What is respiration? and it's a chemical equation, okay? So for those of you guys giving me word equations, unfortunately you will get 0 marks for that, okay? It's chemical, so we have to write out our symbols. Okay, I'm just going to wait for a few more answers. Fantastic. So with our chemical equation, we've got C6H12O6 plus oxygen. Remember, so we get our sugar and our oxygen in the mitochondria. We get the release of carbon dioxide and water. And you can also write energy. You don't have to write energy in the chemical equation because energy is not a chemical, okay? So you don't have to write it, but if you write it, you won't get it wrong. And then it's balanced. Okay, so this is 1 mark. So if you've got that, that's fine. And then just remember your rule of 6s. So 6, 6, and 6. Okay, and that's it. Easy 2 marks. Okay, all right, guys, so it's just gone 10:45, so we're just going to go for a short break. So if you guys come back at 11, and we will move on to talking about plant cells. So grab a drink and have a breather, and I'll see you guys back at 11. Hi everyone, welcome back. So I hope you guys had a refreshing break. So we're just going to continue moving on to our next chapter, and there will be a quiz as well. So do keep your eyes peeled for that. So, okay, so far, basically, we've talked about cells, and we've said that we can divide up cells into two main cell types. Okay, can anyone remind me, what are my two main cell types? I know it feels like I'm saying the same thing again and again, but it will help you guys remember. Two cell types, fantastic. So the two broad cell types are eukaryotic, they have a nucleus, and prokaryotic means they have no nucleus. Okay, so prokaryotic tends to be bacteria, and eukaryotic can be further divided into two main cell types. Okay, they all have a nucleus. What can we divide up eukaryotes into? We've already talked about one of them, and you can see the next one here. Exactly. So eukaryotes can either be animal cells or they can be plant cells. Okay. So we've talked about animal, we've talked about bacteria. So we're going to finish off with talking about plant cells. Okay. So next time you guys go in your back garden or to the park, you guys can have a look at a leaf and you can think about what exactly is inside the cell. So let's just start off. So this is a plant cell. Now you will remember my animal cell was pretty basic. Okay, this was my animal cell. The plant cell is a bit more complex and it's got a lot more going on. So let's just start off with what we know. Okay, now there are, I think, 4 things that are in animal cells and in plant cells. So what is number 1 pointing to? What is this light blue thing? Fantastic. So that is my cytoplasm. So I've got cytoplasm here and I've got cytoplasm here. Okay. What is number 2 pointing to? Exactly. So this is my nucleus and I've got a nucleus over here. Okay. What is number 4 pointing to? So those pink bits. Exactly, that is my mitochondria, where we have aerobic respiration taking place. Okay, and then I can see that I've got an inner layer and an outer layer. So what is my inner layer pointing to? Super, my cell membrane. Okay, guys, so remember I've got a cell membrane here. And I've also got mitochondria here. Okay, so these 4 things, as well as ribosomes, are in plant cells and in animal cells. Okay, I've got a few different things on here which aren't labeled yet. So what is number 5 pointing to? Exactly, so this is the cell wall. Okay, this is not in an animal cell. You do not see a cell wall there. And I've got this really large circle. And it's sort of taking up most of the space in the cell. And this is known as something called a vacuole. Okay, and we'll talk about this. And then the green bits. So these green bits are green for a reason. Okay, does anyone know what they are? Excellent. So these are my chloroplasts. Okay, we'll talk about each of these. So cell wall, vacuole, and chloroplast. What I've written in blue, these are only in a plant cell. Okay, so this is a much more prettier diagram and a bit more detailed. So again, this is 3D and this is 2D. So what you guys can see here is that, well, these are the mitochondria with the squiggly bits. And then I've got my really big hole in the middle, which is my vacuole. And I've got my chloroplasts, which are all the green parts here. And then I've got ribosomes as well. Okay, so these are the extra bits. So the extra parts in a plant cell are vacuole, chloroplast, and a cell wall. So let's talk about the cell wall. So the cell wall was present in a bacterial cell as well. Why do we have a cell wall in the plant cell? It's fantastic. So just like in bacteria, the cell wall is there for support because as you may know that the plant cells are exposed to lots of different environments and they need support to ensure that all of the fluids and everything can stay in place. Okay, otherwise the cell would get damaged. So it gives a lot of support. And the cell wall is made of a specific substance in plant cells. So does anyone know what it is made of? So have a guess. Okay, so I think most of us have got that one right, so I will stop it there. Fantastic. So the cell wall is made up of something called cellulose. Okay, and cellulose is a really, really strong and durable material. Okay, so it's really hard to rip it apart or to break it up. And just so to note, the bacterial cell, the cell wall in the bacterial cell is not made up of cellulose. Okay, this is unique to plants. Okay, next up is my vacuole. Okay, why do you think that there is this big hole or this massive area in the plant cell? And what do you think it's filled with? So we've got to keep the leaf rigid, excellent, and it contains cell sap, fantastic. So basically, inside of this vacuole is it's not just empty, it's got lots of different sugars. Okay, so it's a liquid filled with loads of helpful things, and it's basically got lots of nutrients to help support the plant. Okay, now you guys will know that as animals and as humans, we get our nutrients from the outside, right? Like you eat foods with vitamins and minerals, and we take that in from the outside. Plants, they don't take anything from the outside. Okay, they cannot buy their food. They have to make their own food, and they have to make their own nutrients as well. Okay, for it to stay alive. So that's why they have a vacuole, and that's why we don't, because we don't need to store our own nutrients in this way, but plants do. It also gives it structure. So if you imagine if this thing wasn't there, all of the bits would just be flying around, like the chloroplast would be moving here, and the whole thing would just not, it would be quite unstable. With this vacuole, because it's filled up with a lot of fluid, if you imagine like a balloon inside of it, a balloon that's filled with lots of gas, it's sort of very supportive, and it will just keep it in place. Okay, so this is filled with cell sap. Cell sap is essentially water. Okay, so it's just water, but it's got sugar and it's got mineral ions. Does anyone know of any ions that might be inside? This is just a bonus question. What kind of ions have you guys heard of or do you think might be useful to a plant? You're not sure. Okay, so some ions, things like sodium, magnesium ion. Magnesium is really important and it gets that from the roots actually in the soils. So you guys may have heard of things like fertilizer, which we put in the soil, and that contains lots of nutrients, which then goes up in the plant. Fantastic. Okay, so the pressure in the vacuole presses the cytoplasm against the wall to keep the cell turgid. Okay, now turgid is a really special term that we only use in plants. So you don't say that, you know, like the cells in our body are turgid. This is specifically for plants. So again, if we take the balloon analogy, so if you imagine this is like a box, okay, so just say you've got like a box, okay, and inside you put a balloon that's filled with water. So if the balloon is filled with water, it becomes very tight and firm, right? And because it's so tight and firm, it sort of exerts a pressure on the area around it, because it's very, very strong, and it's just sort of pushing everything away. And that kind of force, what it does is it helps keep the whole cell in place, and it helps keep it compact and what we say turgid. Okay, so it's got enough fluid, and it just keeps it in its place so that the whole cell doesn't burst or collapse or shrivel up. Okay, does anyone have any questions on what I mean by turgid, or are you guys okay? Okay, super. So let's move on. So that is the vacuole, guys. I will be quizzing us on this. So we then got the chloroplast. So what is the point of chloroplast? Okay, so chloroplasts are basically parts of the cell, which, as you guys have rightly said, they absorb sunlight. So this is the chloroplast. Okay, so these are chloroplasts, and basically they contain a pigment inside of them. So as you can see, they're green, right? And they have a pigment, which is just like ink. So if you think about like watercolor paint or any inks, they're made up of pigments, right? So they've got a pigment known as chlorophyll, and this is what makes it green. If there's no chlorophyll, it wouldn't be green. And what the chloroplast is doing is it is absorbing light from the sun. Okay, why does a plant need to do that? Why does it need to absorb sunlight? Fantastic. So it needs sunlight for a process known as photosynthesis. Okay, so again, let's keep things simple. So photo meaning light, and synthesis meaning making. Okay, so we are using light to make food. Okay, and that is the whole point of the chloroplast. So why don't we as humans have chloroplasts? Why in our cells do we not have chloroplasts? Exactly. So it's a very sort of trick question, but we don't make our own food, do we? So we don't need chloroplasts, but plants have to make their own food. They cannot buy their food like us, so they need it. Now, which part of this plant over here do you think has got the most chloroplasts? Where do you think chloroplasts are in this plant? Do you think it's in the stem, in the root, in the flower, in the leaf, at the top? Fantastic. So all of the chloroplasts are at the leaf. Okay, that's why the leaf is bright green, and that's why the roots or the stem are not green, because they don't have chlorophyll. Why is all the chlorophyll at the top, and why would I not put it down here? Why is it better to be up here at the top? Excellent, so that it can be exposed to sunlight to get as much sunlight as possible to absorb the sun. Fantastic. So that is a really common exam question. I remember that always coming up, like why is the chloroplast at the top of the plant or in the leaf? So obviously if the sun is here, it can reach the leaf easily, right? It's not going to be able to dig down into the roots. So there's no point in it keeping it here. So it's mainly the leaves that are doing this process. Yeah, it doesn't really happen in the stem or like in the flower or the petals or anything like that. It's always in the leaf. Okay, so we said photosynthesis happens. So what does photosynthesis do, and what is its equation? So if you guys brainstorm some ideas, what do you guys think is happening in the process of photosynthesis? And I will write down the best answer. So my question is, what is the equation for photosynthesis? What is happening? Super. Okay, I'm getting some really good answers. So, in photosynthesis, the plants use sunlight, water, and carbon dioxide to make glucose and oxygen. Fantastic. So, basically, this is the opposite of respiration. Okay, so we've covered two equations, so I don't want anyone to get mixed up. But put simply, this is the complete opposite. Okay, so what is happening is um, if we just take our plant cell, okay, so this is a leaf over here. Now this leaf is thinking, I need some sugars, okay, because the plant needs energy, right? The plant needs to grow, and therefore it needs energy to do that. Now in order to do that, where does it get energy from? Well, it makes its own sugar, and that is through photosynthesis. So what happens is we have got our sun. Okay, this only happens in the daytime, obviously, when the sun is out. And inside those chloroplasts, okay, we get carbon dioxide. Okay, where does carbon dioxide come from? Fantastic. Carbon dioxide comes from the air. And remember, we breathe out carbon dioxide when we do respiration, right? Carbon dioxide is a waste for us. So basically, CO2 is sitting in the air. Okay, and it goes into the plant. And what happens is the plant uses that carbon dioxide gas and it uses water. Okay, where does it get water from? Where does water come? Exactly, so when it rains. So when it rains, water comes down and the carbon dioxide and the water, they react in the presence of sunlight. Okay, so only in the presence of light do they react, and they form two things. So they form glucose, which is the sugar. Okay, and they also release something else. They release a gas. Okay, they release oxygen. And for them, oxygen is a waste product. So they didn't actually want the oxygen, but they give it out. And that is where oxygen comes from. And that's how we can breathe it in and we can survive. Okay, so the oxygen that the plants give off, we breathe in, and the carbon dioxide that we give out, the plants take it in. Okay, so it's a very balanced cycle, but there are lots of issues because, as you may know, with trees being cut down and things like that, affects the balance of the ecosystem. But anyway, so this is photosynthesis. So we need light energy for photosynthesis to take place. We've got carbon dioxide plus water in the presence of light. Okay, and you must put light energy in the arrow because otherwise you won't get the mark. Gives you glucose and oxygen. So we don't write light on this side, like you don't write light plus carbon dioxide plus water because light is not actually reacting. It's just needed for the reaction to take place. So that's why you always put it at the top in front of the arrow because it's part of the process, but it's not a reactant. Okay, you don't mix in light with this. It's just needed for the reaction to take place. Okay, so guys, just for bonus points, can I have the chemical equation of photosynthesis? Okay, and I'm going to cover up the equation now so you guys can remember it. So what is the chemical equation for photosynthesis? And if I can have it balanced as well, please. And if you can't balance it, that's fine, don't worry. As long as you can give me the just the symbols. Fantastic. Okay, so if you guys are giving me answers, let's just go through this together. So we said in our leaf, we get CO2. Okay, we react it with water from when it rains. In the process of sunlight, okay, you can just write light energy here. So when it's sunny, this is not happening in the dark. We give off two things. So firstly, the plant gets its sugar, and sugar is the same, it's glucose. Okay, glucose is the basic unit of energy. So even next time you have like a sugary drink or cakes or biscuits, they're literally all made up of glucose. This is the simplest sugar. Plus oxygen. The plant doesn't need the oxygen, but we do, and it gives it off and we breathe it in. So this is your basic equation. All right, guys, so we've given ourselves 1 mark, but we want to balance it because it doesn't make sense. So immediately you can see there's 6 carbons here and there's only 1. So don't worry about, this is not chemistry. I would just keep it simple, and it's the rule of 6s. Everything gets a 6, except this one because this has got 6s. Okay, and it will just be balanced. So don't sit there in your exams being like, oh, there's a 6 here, there's a 12 here, and try and work it out. It's just 3 6s, okay, and you'll be done, and we'll get 2 marks. So that is photosynthesis, and that is the process through which plants make their own food. Okay. So guys, just a quick recap on what we have covered before we do a few exam questions. These are our 2 types of eukaryotic cells, okay? And the bits in green are the ones that are only present in the plant cells, and these are both present in all of them, okay? And for some reason they haven't labeled ribosomes, but ribosomes are also in both of them. Does anyone have any questions on this? So for some of you guys, this might be really easy. This is like the basics of cell biology. So if you've got a good understanding of this by the end of the session, it's absolutely fantastic, because this is like really easy marks in the exam and really important, especially if you do biology later on. So yeah, any questions, guys? Okay, let us move on then. Okay, just a bit of a recap actually. So I've just shown you guys a diagram. So tell me, cell membrane, is it present in animal or plant cells or both? So just write down either animal or plant, or give me both. Fantastic, both. Okay, cytoplasm. Let's whiz through this. Super, both. Nucleus? Both, excellent. Mitochondria? Both, fantastic. Okay, cell wall, animal or plant or both? Yay, well done, guys. I'm really happy. So it's in plants only. Chloroplast? Fantastic, it is in plants only. And vacuole, it is in plants only, okay? And ribosomes that make protein are in both. Okay, excellent. So that is the basics. Okay, so let's do an exam question now. Okay, so this question is about cell structures. Draw 1 line from each cell structure to the type of cell where the structure is found. Okay, so let's start off with the nucleus. So if I just say A, B, or C, if you guys want to just vote on the poll, which one do you think it is? So for nucleus. So remember the definitions of eukaryotic, prokaryotic that we said in the beginning. Those are like the main ways to divide up cells. Okay, I'm getting a few mixed answers. This is a bit of a confusing question. Okay, and I will stop you guys there. So most of us have said C. So nuclei are present in animal and in plant cells, right? We just talked about that here. There's a nucleus here and there's a nucleus here. Animal and plant cells are both eukaryotic. They contain a nucleus. So the correct answer would therefore be C. Okay, so what about a permanent vacuole? A, B, or C for my permanent vacuole. By the way, guys, vacuole and permanent vacuole are the same things. They just write permanent sometimes because it is always there. Fantastic. So this one is B. Permanent vacuole is present in plant cells only. Okay, and therefore, the last part of this question, plasmid, what do we think is plasmid. So it's not just guesswork, it could still be A, B, or C. Which one do you think? Where is the plasmid present? Excellent. So the plasmid is present only in the prokaryotic cells. And just to really drum this in, what is an example of a prokaryotic cell? Fantastic. So prokaryotic cells are bacteria. Okay, super. So I think these questions are actually a little bit easier. So I think I will leave them because I think we're doing really well. So let's have a look at this one. Plants absorb light to photosynthesize. What is the correct word equation for photosynthesis? So these questions are okay. Later on, and especially over the course of the year, we will be doing like experimental questions and questions that really challenge you to think. And the way that they come up in the exam is slightly different. So we will be doing a range of questions. So which one is the equation for photosynthesis? So just make sure you read the question carefully. Okay, fantastic, guys. So the correct answer here is it's D. Water plus carbon dioxide gives me oxygen and glucose. So remember, guys, photosynthesis, we're using light to make food. Okay, what is my food? My food is glucose. So if I'm making glucose, glucose has to be on this side, right? So there's no glucose here, that's gone. There's no glucose here, that's gone. So I know it has to be only one of these two. And remember we said that oxygen is given out in photosynthesis, okay? The plants give us the oxygen that we need. So I already know it's not this, okay? Because I know I need oxygen. So this is my correct answer over here. Okay, any questions, guys, so far? Are you guys finding the pace of this session too fast, too slow, or is it okay? Because I'm really happy to slow down or speed up. Just let me know. Okay, you guys are happy. Great. So the diagram below shows a plant cell. In which part of a plant would you find this type of cell? Where would we find this type of cell? So we know that this is a cell in the plant, right? But the question is, where in the plant would you find it? So the part of the plant that this would actually be in is, because what we can see is that it is loaded with these bits, okay? So this is black and white because it's from the exams, but it's labeled as chloroplasts. And remember we said that chloroplasts are there for photosynthesis, and in our plant, the chloroplast will sit at in the leaves, okay, so that the sun can directly get it. So the part of the plant, obviously other options are like your stem, your root, the flower. That doesn't make any sense, right? So it has to be in the leaf. Okay, some of you guys are giving me answers like palisade layer in the waxy cuticle. So those are not parts of the plant, those are parts of a leaf. Okay, when you take, we'll do this when we talk about photosynthesis, but when you take a cross section of the leaf, you see layers like waxy cuticle, palisade cell. Those are parts of the leaf, they're not part of the plant. Okay, so this is a simple question and it's in the leaf. So I hope that makes sense. Okay, give the function of the nucleus. Fantastic. So we can say it contains the DNA. And it controls the activities of the cell. Yeah, those are two things that we can say. And you guys have all said that. Okay. Give the function of the chloroplast. Fantastic, to absorb sunlight. And what is the key term that we want to use? Fantastic, for photosynthesis. So that would be your best answer. Okay, so the chloroplasts are there for photosynthesis. So when we do exam questions, we want to say keywords. Okay, you don't want to be vague and be like, oh, to get light or things like that. You want to use key terms. So we're absorbing sunlight for a process known as photosynthesis. Okay, and some of you are saying release energy. That is for mitochondria. Okay, and we'll just do a bit of a more challenging question. Okay, so tick one box in each row to show whether the statement is true for photosynthesis or for respiration. Okay, so carbon dioxide is produced. So do you guys think that's photosynthesis or respiration? So if you guys just write P or R for when is carbon dioxide produced? Fantastic, respiration. Okay, when is light needed? P or R? Fantastic, photosynthesis. Okay, I have been talking about the sun a lot, and it occurs in plants and animals. So respiration occurs in both plants and animals, okay? Because remember, both plants and animals, we all need to respire to release energy, but photosynthesis is not in animals, so we know it can't be that one. And finally, oxygen is produced. When do we produce oxygen? Fantastic. So oxygen is produced in photosynthesis, okay, as a waste product, and it is the air, is the oxygen that we breathe in. Okay, respiration, on the other hand, uses up the oxygen. Okay, does anyone have any questions so far, guys? We've covered eukaryotic and prokaryotic cells in a nutshell, and I want to move on to something a little bit more, the next part of this topic. Okay, please just ask any questions as soon as they come up. So before we do that, guys, you guys are doing really well. So I just want to do a quick quiz just to make sure that we are all on the same page. So if you guys are able to log into Kahoot, kahoot.it, and this is the pin. And if you guys don't use your names, but just log in with a new word or something that you've learnt in today's biology session so far. Okay, some of us knew all the words already. Okay, fantastic. Eukaryotes, plasmid. I think that's my favorite one. Okay, just give a couple more seconds till we all log on. So guys, this quiz is not that hard. So I want to see who can get there as quick as possible. So keep your fingers on the trackpad as soon as a question comes up. Okay, I think we'll get started. Okay, so these are examples of specialized cells, which we'll talk about. Fantastic. This is a sperm cell. All right, bacteria is in the lead. Super, and we have got a red blood cell. Okay, what type of cell is this? Okay, guys, tricky question. We are split between animal and plant cells. So for those of you that said plant cells, do you guys see a vacuole on here? Do we see chloroplasts? Do we see a cell wall? Okay, so this is actually a very typical animal cell. So this is mitochondria, and these are ribosomes, and these are also this is a little bit more complicated, but these are known as endoplasmic reticulum, but they are part of the ribosomes. So this is not a plant cell, and the most obvious feature is because there are no chloroplasts or vacuole, but tricky picture. Okay, and plasmid is now in the lead. What is the purpose of mitochondria? Fantastic. So mitochondria are there to produce energy. They do not produce food. We use food to release that energy, and they don't store it. Energy is actually stored in other places, and it's stored usually as fats or as lipids. Fantastic. So the cell wall provides support and protection. Great. So what are the reactants of cellular respiration? Excellent. So remember, we are using glucose and oxygen. All of these options don't really make sense. So that's quite an easy one. Okay, so respiration actually produces something called ATP. So well done if you got this a little bit of more A-level biology, but the way that we store energy is in a form called ATP. And yeah, it's basically another word for stored energy. So respiration doesn't create cells, guys. Cells are created by cell division. So the products of aerobic respiration are water and carbon dioxide. Fantastic. Excellent, well done guys. So a lot of you got 4 marks or close to 4 marks, so that is really, really impressive. So I think we have really consolidated what we've just learned. So we're just going to move on to our next bit that we are looking at. Okay, and that is basically the microscope. Okay, so I've got a few objects on here. What do all of these objects have in common? Excellent. So yes, they're all made of glass, but more importantly, they all have a lens. And they all have a lens because they're all used for magnification. Okay, so all of these things are used to magnify, i.e. to make something bigger. Okay, you've got your glasses and a microscope, a lens here, and you've got an actual microscope. Okay, so when we talk about the microscope, we know that a microscope basically magnifies the image, an object, and the picture that you see under a microscope is called the image. So are you guys doing this topic at school? Have you been using microscopes at the moment? Okay, some of you yes, some of you no. So basically with the microscope, what happens is let's say you want to look at like a piece of like a leaf, okay? So what you do is you take that leaf, you just take a really small part of it and then you put it under a glass, okay? And then you put that glass just underneath here and then you look at it through the microscope. So you put your eye in through here, okay? And basically you see a magnified version of that small leaf. We call that the image, okay? Because obviously it's not real what you're looking at, it is an image of the leaf. And a microscope has got a lens. So inside here is a lens, which makes the object bigger. It's just like your glasses. There is a glass there, which makes it much bigger. And microscopes are obviously really important because most things are invisible to the naked eye. So all the layers of the leaf, many bacteria, and really, really small organisms, you cannot see them. So microscopes have really helped us learn a lot. Now, when we talk about microscopes, there are two things that we look for in a microscope. So we can talk about its magnification, and we talk about its resolution. Okay, so with light microscopes, so these are the ones that you use in school. I know some of you haven't, but this is typically what they have in school. So light microscopes are used when basically a small amount of magnification is okay and just a small amount of resolution. So magnification is basically how big and resolution is how clear. So what can you guys see as a difference between these two sheep? Like the image of the two sheep. Exactly, so one of the sheep, this sheep is clear and this one is blurry, right? So their size is the same, so their magnification is the same. They're the same size, like the ears are the same size, the face is the same size, but the clarity of which you can see it, this one is clear and this one is very blurry. So with a light microscope, the resolution tends to be quite low because it's quite a cheap instrument. So you can't really see very clearly and the magnification is not that high. So you can see maybe like 50 times, but it's not that high. So magnification is how large it is, okay, and resolution is how clear the image is. So this is basically your light microscope here. And I hope that you guys do get to use these a little bit more, but let's just label this together. So at the top here, I've got my eyepiece. Okay, and this is basically what you use to look through it. Okay, I've got my eyepiece here. What is the next bit that I can label? So what is the round thing? And what is this whole round thing here? Fantastic. So that is my lens, and that lens is basically magnifying it and making it bigger. Okay, does anyone know what is sitting over here? So there's actually a mirror over here, and the mirror is just there for reflection. So whatever you put on there, it reflects it back up so you can see it. If there was no mirror, you wouldn't be able to. And then this knob over here, if you've ever used a microscope, you know that you swivel it up and down. And basically what that does is it changes the focus. So it just moves the lenses around onto different parts of your plant or whatever you're looking at. Okay, and it just helps you see it a little bit more clearly. If you do it in school, you'll just be fiddling around with it, and then all of a sudden you can see your image. This is the stage. The stage is where you put the slide on or whatever you're looking at. And those are basically the parts of your microscope. Does anyone have any questions on this? You don't need to know too much about microscopes, but it's just good to have a basic understanding, and we will talk about the calculations as well. So that is my light microscope. We have also got something known as an electron microscope. So the electron microscope is totally different in that it is much larger. So in microscopes, we have got light and we have got electron. So light microscopes, you can carry them around, you've got them at school. Electron microscopes are much larger and you can see them over here. They're used in like research labs and in the industry. Now the way that they work is they don't use light, they actually use a beam of electrons. So there's actually a beam of electrons that flies through. And they have a much higher magnification of up to 1 million. So the max magnification of a light microscope is usually around 50 times, but electron microscope is times 1 million. So you can really, really look at the details. The only difference is that they cannot be used for living cells because the samples are put in a vacuum. So in the light microscope, you can put like a leaf, you can put bacteria that is alive, but in the electron microscope, you can't because it will end up dying. And they're very large and they're quite hard to move, so they're quite bulky. And obviously you have to keep them in the same place, whereas a light microscope, you can carry it around. Okay, so what is the point of our microscope? So with our microscopes, we can actually calculate the magnification. And the magnification, there is an equation for this. We use two things. We use the size of the image and we divide it by the actual size. That will allow us to calculate how magnified something is. Okay, so magnification is equal to size of image over actual size. Okay, so what can happen is sometimes you might see that your image is really, really big, but actually when you look at it, it's so tiny. So that will allow us, by comparing these two, we can see how many times something is magnified. These questions, they love to ask this in exams and it always comes up. So the main thing I would warn you guys about, just to remember, is your units, okay? So make sure that you keep the units the same. And in microscopes, we use very, very small units. So we know that 1 centimeter is equal to 10 millimeters, but 1 centimeter is equal to 1000 Does anyone know what this stands for, this UM? Excellent. So this stands for micrometers. Okay, so those of you that are saying nanometers, that's nm, okay, and that is different. We're talking about micrometers here. So 1 centimeter is equal to 1000 micrometers. Okay, So let's have a look at a question. Okay, so let's have a look over here. So the actual size of this cell is 0.1 millimeters. What is the magnification? Okay, and you can use the ruler to measure it. And here is my equation over here. So have a go at this. So how many times has this cell been magnified? Okay, and I think we will just do a poll. Okay, so I'll give you guys a couple more minutes to answer that one, a couple more seconds. Okay, so most of us have gone for C, okay, and some of us have said A as well. So let's just talk about that one. So my equation is here, right? It's magnification is the size of the image over the actual size. So we already said in the question that the actual size is 0.1 millimeters. So this is 0.1 millimeters. And then the size of my image has come up to 6 centimeters. Okay, now remember, you want everything to be in the same units. So 6 centimeters in millimeters is what? Exactly, it's 60 millimeters, right? So 1 centimeter is equal to 10 millimeters. So 6 centimeters is equal to 60 millimeters. Okay, so we know magnification is size of image over actual size. So that will be 60 millimeters over 0.1 millimeters. Okay, and that will give me 600. So you know that your magnification is 600 times. So this small cell has actually been magnified 600 times underneath that microscope, which is quite a lot. Okay, does anyone not understand what I did there? Okay, let's have a go at another one. Okay, a few mixed answers on this. We're split between B and D. Okay, so half of you guys have said B and half of you have said D. So let's just go through this one. So just use our equation. So the actual size of the cell they've told us is 0.2 millimeters, right? So this is 0.2. And if we measure the size of the image, so it's 4 centimeters, right? So 4 centimeters, I have to convert that into millimeters. So 1 centimeter is equal to 10 millimeters. So 4 centimeters, I times this by 4, times this by 4, that will be 40 millimeters. Okay, so 40 millimeters divided by 0.2 millimeters will give me 200. Okay, so my magnification is 200 times. So my correct answer there was D, in fact. Okay, so that is basically using the equation for magnification. Okay, so do try and remember this: size of image divided by your actual size, because sometimes they won't give it to you in the exam. Okay, and just a quick recap between our microscopes. So if we were to compare a light to an electron, which one is bigger, the light or the electron microscope? Fantastic. So the light one is smaller. The electron microscope is larger. Okay, what do we think about cost? Which one is cheaper, light or electron? Fantastic. So the light microscope is cheaper and the electron microscope is more expensive. Okay, the magnification, which one can magnify more? Exactly. So my electron microscope has got a much higher magnification than my light microscope, which is a bit lower. Okay, and then my resolution. Fantastic. So my resolution of the light microscope is much lower and the resolution of my electron microscope is much higher. So it can see things more clearly and it can see it much more larger. Okay, and then in terms of my specimen type, so can I use living objects or dead objects in the light microscope? Fantastic. So in the light microscope, you can use living organisms. So you can just take like a piece of leaf and put it in. In the electron microscope, the organisms are dead because basically it's using electrons, it's going through a vacuum. So you can't put something alive in there because it will end up dying. So basically light microscopes, they are cheaper, but they're not as good with magnification or resolution. Electron microscopes are more expensive, but overall you can see better and see much larger through it. Okay, so let's do a little bit of exam practice. So use the scale to measure the length YZ on the photograph. So this is a 1 marker. So if you had this on a piece of paper, it would be a bit easier to do, but you can just have a look. So basically they've put a ruler there and they're asking you to measure this bit. So this is 0 and where Y is, and this is Z. So if you look on your ruler, it's 80, this is 85, right? Super, so this is, and the scale is in millimeters, just be careful. So this is 85 millimeters. Okay, so now they're saying the photograph shows the root magnified 100 times. Calculate the actual length YZ. Okay, and the image size, we've just seen it on the previous slide, is 85 millimeters. So have a go, guys. I won't reveal the equation because we have talked about it. So without looking at the equation, have a go. So let's start working this out. So we know that magnification is equal to my size of my image over my actual size. Okay, so they're asking me to calculate the actual size. So I have to reverse the equation in this case. I'm not working out magnification, I'm working out actual size. So what's my size of my image? They've given us that, we worked it out. Exactly, my size of my image is 85 millimeters, yeah? That's what we saw in the previous slide. And it has been magnified 100 times. So all I have to do is 85 divided by 100, which will give me 0.85 millimeters. Okay, so some of you guys, I think, multiplied it and end up getting 8,500. Now, if you think about it, that doesn't make any sense, right? Because the image size is only 85 millimeters. So the actual length has to be much smaller, okay? Because you're putting it under a microscope. So the actual length of this is not going to be 8,500. It has to be much smaller. Okay, so just think about that when you get your answer. Does it actually make sense? It can't be so big. It has to be smaller. So this number has to be bigger than this one, yeah? So it's 0.85 millimeters. Does anyone have any questions? Okay, super. Well done, guys. So it's just gone 12 o'clock. So I think it's time that we all get some lunch. So you've all done really, really well. So it's 12:02. So if you guys just go for a quick half an hour break, and if we come back at 12:35, okay, so grab some lunch and leave any questions in the chat. And we will move on to our next topic after the break. So I'll see you guys then. Hi everyone, welcome back. So I hope you all had an enjoyable lunch break and managed to grab something to eat. So we've not got long left, we've got just under an hour and a half. So we're going to move on to talking about two other parts of today. So we're going to talk about specialized cells now. Okay, so this came up in the quiz, so I think you guys have got a good understanding, but we've talked about cells in general, so animal cells and plant cells, but divided up into these, there are many different types of cell. Okay, that's just the general type. But we've got many different types of cells which have a specific role to perform, and they only do that job, and they are suited for that. So they've got adaptations and features that help them do that job most effectively. So do you guys know any specialized cells? There's a few that always come up. What type of cells do you know that are special? We have named a few of them today. Fantastic. So sperm cell, muscle cell, root hair cell. Yep, red blood cells, fantastic. And xylem and phloem as well in plants. Okay, so there are loads of cells, and we're going to talk about a few select ones of these in animals and then in plants. Okay, so here are my red blood cells. This here is a sperm cell, and this here is a nerve cell. Okay, so red blood cells, what is the job of a red blood cell? Can you guys tell me? Why do we have red blood cells? So you know that if you get a cut, you will see that blood comes out and that blood is red, and that is because we have red blood cells inside of our blood. And the main thing that the red blood cells do is, yes, they travel all around the body in our vessels, but they transport a gas. Okay, so they transport oxygen around the body. Why do we need oxygen? We've talked about this earlier today. What do we use oxygen for? What is the name of that process that we need oxygen for? And it is happening in a specific part of our cells. Excellent. So we need oxygen for respiration, okay, so that we can release energy. Otherwise, without it, we wouldn't be able to do any of our activities. So the red blood cells, they carry oxygen. Now, if you look at this, your typical animal cell looks like this, right? How is the red blood cell different? There are a few key factors. What makes it different? So what can you see that is different between this normal animal cell and a red blood cell? Great. So the first thing is that they contain hemoglobin, which carries oxygen molecules. So inside this red blood cell, it's made up of mainly one component, and that is hemoglobin. Okay, and in medicine, we write this as HB. Okay, so hemoglobin is what binds to the oxygen molecules, and it carries them along. So in like your muscle cell or in your liver cell or in your eyes or in your hair, you don't have hemoglobin, okay, because they're not binding to oxygen. Your muscle is not doing that. So hemoglobin only occurs in the red blood cell, okay? And hemoglobin is a protein. This is why we need ribosomes. It's a special protein that can bind to the oxygen, and it will carry O2 around the body. What other things can you see in this red blood cell that are different to our normal cells? Fantastic. So the second thing is that they don't have a nucleus. Okay, so this is probably one of the only cells in the body that doesn't have a nucleus. We said that all eukaryotic cells, they do have a nucleus, but the red blood cells, they don't. Okay, there is no nucleus. Now, what that means is if you get rid of this nucleus, it means that there is more room to carry around oxygen. So instead of a bulky nucleus clogging it all up, it just means that the cell is free and it can pack in as many O2 molecules as possible. Okay, so this feature is a specialization, and that's why this is a specialized cell, because it is adapted to performing its function. Okay, its function is to carry O2, and it is adapted to doing that. Okay, there's one other thing to do with the shape of this cell that makes it specialized. Does anyone know what that is or what it's called? Excellent. So the last thing is that if you look at this shape of this cell, it sort of looks like a donut, doesn't it? If you were to cut it in half, this kind of looks a bit like a Krispy Kreme donut. So you've got a hollow bit in the middle and you've got this ring on top. Now, the term that we describe this is known as biconcave. Okay, bi meaning two, and concave meaning it's got this hollow shape. And what that means is that it's just got much more room to carry the oxygen. So if it was a flat disk, the oxygen can only go over here. Okay, now what we've done is we have made the disc dipped. Okay, so there's kind of in a bit. Can you see there's now all this extra room over here to carry the oxygen? So it just gives it much more room to carry it, and it means that more oxygen can be carried at any one point in time. Okay, so we say that they are flat disc shaped, they are biconcave, and that gives them a large surface area. So because they've now got more surface to which to carry the oxygen, you can carry more O2 at any one point. Okay, so those are our red blood cells. Moving on to our nerve cells. Okay, so this is a nerve cell over here, and nerve cells transmit electrical signals in the body. So this is our man over here, and we will do a whole topic on the nervous system. But in our nervous system, there are two parts. So we've got our central nervous system. Okay, which is made up of the brain, that's over here, and the spinal cord, which is running through here. And then we've got our peripheral nervous system. Okay, which is all of the nerves that are going through. Now for these to work, okay, we need millions and millions of nerves. So if you want to pick up your pen, or if you want to read something or get up, all of these are nerve impulses that tell your brain and your body what to do, okay? So nerve cells are very, very vital for our functioning, and they look like this. So they have three main ways that they are suited to their function. Okay, can anyone name any by looking at this diagram here? So they are very long, excellent. Okay, really good answers, guys. So basically, the first thing is this is my nerve cell over here. Now, the main thing to remember is that they are very, very thin. Okay, and they're also very long. So they are about 1 meter long. So 1 meter is huge. Okay, they can run all the way from the top of the brain right to your foot. So because they are long, they can easily transmit impulses up and down the body. Okay, so these are electrical impulses that can travel up and down the body at a very, very quick rate. So when we describe our nerve cell, we can divide it up into different parts. And basically what you can see here is this is my nucleus. Okay, this bit over here is known as my axon. Okay, and these, you can see that there are loads of different spiky bits. Okay, and we call these the dendrites. Okay, and all of these parts of the nerve cell help it to carry impulses because that is its job. So the first thing that we can say is that the axon is very long and it's very thin, allowing it to transmit impulses. The next thing that we say is that they have got branched connections, as you guys have rightly said. So this nerve cell is not sitting on its own. It's got all of these branches, and then these branches then go and connect to another nerve cell. Okay, so this one connects to a nerve cell here. This one will connect to another one. And this one will also connect to another one. Okay, so this might be a nerve cell. This might go to your eye, and this might go to your leg, and this might go to your hand. So they're all connected to each other so that your whole body can communicate whenever you need to do something. Okay, so we call these bits the dendrites. And then finally, what we can see here is that there are these blobs around the axon. Does anyone know what this is called or what this is made up of? What are these yellow pieces on it? Okay, some of you are saying mitochondria. Try again. So what we have already said is that, let's just go back to this diagram, which I think is a little bit better. So what we have said is that this nerve impulse is carrying something to the brain, okay, from the brain to the body. So it's going to carry that impulse all the way along. Now, actually, what can happen is this can be quite a slow process. This is 1 meter long, and by the time it gets all the way from your brain right to your leg, it can take a bit of time. So the nerves are very clever, and instead what they have developed is these circular bits around it. Okay, and we call these circular bits a myelin sheath. Okay, and what this myelin sheath does is it allows the nerve impulse to jump across it instead. So what happens is instead of just going straight through like this, the impulse jumps and it goes up, down, like this. And this is much quicker, okay? Rather than it going all the way, dragging along through here, it just bounces in between the myelin sheath. And this fatty sheath speeds up the impulse. Okay, so that is the role of the fatty sheath. So it's made of myelin, okay, and it increases the speed at which the nerve impulses can travel. Okay, does anyone have any questions on that? Okay, so we've talked about our red blood cell and we have talked about our nerve cell. Okay, so let's move on to our sperm cell. So the answer is actually on the slide, so I don't want to give it away already. So let's just test ourselves. So I'm going to draw my sperm cell over here. So guys, I know we're all a little bit sleepy. We've had our lunch, but this is important. Okay, so this is my sperm cell over here. What is the point of a sperm cell? What does it do? Why do we need a sperm cell? Exactly. So the sperm cell is there to fertilize the egg. So now that we know its role, we want to think about, well, how is it suited to its role? So if it's there to fertilize the egg, when we look at the shape of it, what things does it have that will suit it to doing that? What can you see on this diagram? Excellent. So it's got a tail. Now we can divide up the cell into 3 parts, okay? So this bit over here is my tail. This bit over here is my body. And this bit over here is my head. Okay, so I've got a tail, body, and a head. Now, each of these is very, very specialized and suited to performing its role. Now, why would it have a tail? Well, we know that the tail is there to allow it to swim. If you imagine if it was just this blob here, it wouldn't be able to do anything. So it's got a very long tail to allow it to swim very quickly to reach the egg, okay? It's then got a body. Now the body has got something in it. So it's not just, you know, empty. There is something very special inside the body. Does anyone know what that is? What is inside the body? Fantastic. So in my body, I've got many mitochondria. Okay, and what is the point of mitochondria? As we know, we said this lots and lots of times, mitochondria are like my battery, okay? And they are providing energy. Now this sperm is not going to want to swim on its own if it doesn't have enough energy, okay? It won't be bothered. So that's why the mitochondria have got lots of energy to help it swim. Okay, so tail to help it swim quickly, mitochondria to give it the energy, and then we've got our head. So the head of the nucleus, the head of the sperm cell, it's got a nucleus, but it's also got something else inside here. Does anyone know what the head has got? Now, if you imagine the head is the main part that is going to reach the egg. So this bit over here is my egg. Okay, now the sperm is trying to get through to that egg and break through it and get inside. So what do you think it might have inside? Excellent. So it has basically got enzymes. Okay, now what do enzymes do? Enzymes help in speeding up a reaction and in helping break things down. So what happens is as soon as the head reaches the egg, it releases these enzymes and it helps break the wall of the egg down so that the sperm can get through. Now, we give this part of the head a very specific name. It's called the acrosome. Okay, and acrosome basically means that it's got enzymes. So this bit here is the acrosome. And it contains enzymes to help the sperm swim to the egg. Okay, so let's just try an exam question now. So cells can be specialized for a particular job. The diagram shows the structure of a human sperm cell. Describe how the long tail and the mitochondria help the sperm to do its job. So guys, this is a 4-mark question. Okay, now when it's 4 marks, that means 2 marks here and 2 marks here. So I'll give you guys a couple of minutes. I want a very full detailed answer of how you would write this in the exam, okay? And then I'll give you guys some feedback. So have a go. Let's start off with long tail. And they've given you a diagram to help. And this is a describe question. So remember, there are different types of questions. There's explain, there's suggest, this is describe. So you need to give a really thorough description of what the long tail does. So just wait for a few answers, but you guys are on the right track. So well done. Fantastic. So we can say that the long tail allows the sperm to swim. Okay, so that's 1 point. So the mark scheme says allows the sperm to swim. Okay, so swim is the key word here. Allows the sperm to swim. And the second thing is that, yes, it allows it to swim, but what else can we say? The other point is that it swims towards the egg. Okay, because can you see the question is saying how is it helping it to do its job? So its job is to break through to the egg, right? So you must say that. So it allows the sperm to swim towards the egg. Okay, so you have to say both of these to get 2 marks. Okay, let's have a go at mitochondria now. So we've said that mitochondria was sitting in the body, right? So mitochondria release energy. Okay, so that's one point. We know it releases energy, but what do we do with the energy? So you can say to allow the sperm to swim, or you can say to allow the sperm to move. Okay, so can you guys see that? You have to describe what it does and then how that helps it does its job. Its job is to swim and to move to the egg. So you must say that for both of the points. Okay, does anyone have any questions on that one? I think most of you would have got both marks for each of them. And those of you guys that are mentioning the acrosome and the enzyme, so remember that is sitting here in the head, so you won't get any marks for that because they're not asking you, they're just asking about the long tail and the mitochondria. They're not saying what is the point of the enzymes. Okay, super. Okay, so let's move on. So use words from the box to label parts A and B. Okay, what do you guys think? What is part A? So this is fairly easy, hopefully. Excellent. So that is my cytoplasm. So remember, all cells have got a cytoplasm over here, and B would be my excellent. That is my nucleus sitting in the head. And just to be clear, this bit here is the acrosome. Okay, this is the mitochondria, and this is the tail, which they have kindly labeled for us. Okay, I think this is quite easy. Okay, so sperm cells have many mitochondria. Why do sperm cells need many mitochondria? Okay, what do you guys think? A, B, or C? Excellent. So they're not because they're involved in fertilization, not because they're produced in large numbers, but because they need a lot of energy to swim so that they can release energy and they can swim much more quickly towards the egg. All right, guys, so we talked about our animal cells. The next type of cell which I want to cover is the root hair cell, okay? So root hair cells occur in plants. So we've talked a little bit about our plant. Okay, this is our leaf over here, and this is my stem. This may be the flower, and then going down into the soil are my roots. Okay, and if we were to take a cross section and look at a really small part of this root, this is actually what it looks like. Okay, so it's got a very, very unique shape, and we're going to talk a little bit about this. So just ignore the text and have a listen. So this is the whole root hair cell over here. And what we can see is that there is this long bit that's sticking out and then the main part of the cell. And this is the soil over here. So the main specialization are these extension bits, okay? And these are the root hairs. Why do you think we have these extensions? Why can't it just end over here? So what is the point of those pointy bits? Because in theory, the root hair cells, they could just be sitting at the bottom and they could just be sitting as blocks. Okay, but instead they have this extension that really goes deep into the roots, each of the cells. So why should they have that? Exactly, well done. So a lot of you guys said to collect water and to get nutrients from the soil. So basically, the point of the roots, as we know, is the roots, they collect water and they connect minerals from the soil. That's why we put fertilizers in our soil, and they take them up to the rest of the plant, okay, in order to be used by the leaf and the flower. That's the whole point, because the plant needs water. Now, what would happen is if this was just chilling in the soil, okay, the soil is just over here, it wouldn't really be able to do anything because the soil is quite dry. So instead, it's extended its way all the way out so that it can really draw in and absorb all of the water and all of the minerals in the soil, okay? So the extensions help increase its surface area so that it can really widen its roots and get into the soil to absorb the water and the minerals. And that is basically the main specialization of the root hair cell. So you always want to think about, what is the role of it? Well, the role is to absorb water and to absorb ions. How does it do that? With the root hair cell extensions. Okay, and they poke out into the soil. Does anyone have any questions on that? Okay, I think we'll move on then. So let's have a look at a question. So the drawing shows part of a root hair cell. Okay, use words from the list to label the parts of the root hair cell. Okay, so this one doesn't look too bad. So let's have a go. So what do we think this top label is? Fantastic. So remember, all plant cells, we said that they all have a cell wall. Okay, so this is my cell wall over here on the outside. What is my next label? What is this inner label over here? Great, so my inner one will be my cell membrane. Fantastic. And then what have I got here, this bit here? Super, so that is my cytoplasm. Okay, and then finally my last label here, which is this big massive hole. Excellent, that is my vacuole. So remember, that is basically like the balloon with the water filled inside, okay? It contains all the cell sap and the nutrients. Okay, guys, so we just got a couple more cells before we move on, okay? So after our root hair cells, we have got the actual root itself, okay? So when we look at the roots, okay, I'm just going to be drawing my leaf diagram again. Okay, so as you know, this is the leaf here, and this is the flower. Now, part of this stem, it needs to carry glucose, and it needs to carry sugars and water and mineral ions all up through the plant, okay, because the flower needs it, the leaf needs it, and so do the roots at the bottom, okay. So how does the plant carry all of that stuff up and down? Well, the way it does that is through two different tubes, okay. So inside our stem, we have got two different tubes, and we'll talk about them now. Okay, so there's quite a lot of text, but let's just have a look at the picture. So xylem cells form long tubes running along the roots and stems of the plant. So inside our stem, you will see that if you were to look under a microscope, there are tubes like this. And basically what this tube does is it carries water and it carries minerals right from the base of the plant and takes it up, okay? Because all of the water we get from the soil and all of the mineral ions we get from the soil as well. So what this xylem does is it carries the water and the minerals up. Now it is specialized, okay? This doesn't look like a normal cell. So can anyone tell me what special features it has? How does it look different to your normal plant cell? So it has less structures inside. Fantastic. It forms lining and builds a tube. Excellent. So guys, what you can see here is that there's nothing in there, right? Like there's no nucleus, there's no chloroplast, there's no vacuole. You can't see any of that. All you can see is a tube. And very interestingly, this tube is dead. Okay, so this is a dead, made up of dead cells that have literally got no content. Okay, so xylem tubes are made up of cells that have died and they have no end walls and no content. So basically, it's just like a hollow tube. If you think of a straw, okay, it is literally a straw, which just allows the plant to just suck up water and carry it to the plant. Okay, there is nothing inside it, and it is not even alive. It's basically dead cells. Now, these bits over here, they look quite thick, and these bits on the sides are made up of a special substance known as lignin or linin, and this is basically there to strengthen it. It's sort of like cellulose in the normal plant cells, but it's for the xylem. Does anyone have any questions on that? Okay, so what we can see here is that this is the xylem. So this is one tube that is present in the plant. And then we've got another tube, okay? And the other tube is known as the phloem. Okay, so xylem is carrying water and it's carrying mineral ions. Now, what about the sugars? We need something to carry sugars. So that is where the phloem comes in, okay? And we've just got a really good question which says, why is the xylem dead? The reason it's dead is because if it was alive, it would be packed with loads of cells and it wouldn't be able to pull up the water. Okay, if you imagine if there was like chloroplast and vacuole, everything chilling in here, there would be no space for the water. So that's why it's made up of dead cells, okay? And it's got lignin to help support it, okay? Otherwise it would collapse when it was filled with water. So this is carrying water. Now phloem, on the other hand, doesn't just need, water can only go up, right? Because we need to carry it up to the plant. But phloem carries the sugars. Now the sugars are actually made in the leaf, okay? And they need to go down, or they may need to go up. So they need to go up and down. So we need a bit more of a complex process. It's not just going to be pulled up this way. Sugars can go up and down. So the phloem is actually much more complicated, okay? And this is where things get a little bit tricky, but I will explain this to you guys. So there's a few parts of this phloem cell. The first one are these cells here. Okay, and these are known as our companion cells. So what do you guys understand by the word companion? Okay, a companion is just like a friend, right? Or it's there to work together or to be supportive, exactly. So basically these companion cells are just there for support to help move the sugars up and down. So what we have is we have got these companion cells and we've also got these plates in between, and these are known as sieve plates. So if in your kitchen you've ever used a sieve to drain out anything, you will know that it just allows some pieces to go through and not everything. So the sieve plates only allow the sugar to go through and they control the amount of sugar going through. So it's not just a whole pile of sugar that's going up and down. It's a very controlled movement, and it only allows some sugars to get through. Now, the process by which phloem cells move the sugar is known as translocation. Okay, and trans is move, and location is the place. So basically, the sugar is moving, and it's quite a bulk movement of sugar. So translocation does not happen in the xylem; it only happens in the phloem. So if we just go through this, so phloem cells carry the sugar made in photosynthesis from the leaves. Remember, it happens in the leaf, and it goes to all of the other plants. Okay, and we call this process translocation. And either we use the sugar straight away in the plant, or it can be stored. So you'll know plants like potatoes, they've got a lot of storage, and carrots and things like that. Okay, now unlike the xylem cells, phloem cells are alive. And they have fewer cell organelles than many other types of cell, which allows the sugar to travel easily, but they have got more than the xylem, okay? And phloem cells have specialized end wall called sieve plates that have holes in them to just allow the controlled movement of the sugar to come through. Okay, and phloem cells are arranged with xylem cells to form bundles. So next time you actually look at a leaf, okay, you will see that on the leaf there are like those really, really skinny lines, okay, that are sometimes not even obvious, they're so faint. Those are actually the lines of the xylem and the phloem, okay, so you can understand how tiny and microscopic they are, but they carry on into the stem and the root and the rest of the plant, but they're really, really very narrow. Okay, so just a quick recap, so we are all on the same page. What is this a diagram of? What is this showing me? What type of cell? Fantastic. So this is xylem. Okay, and what do I have on the right? Super. This is my phloem. Okay, what is the xylem carrying? Fantastic. So my xylem is carrying water. Okay, and it is carrying mineral ions. Okay, what is my phloem carrying? Super. So my phloem is carrying sugars. Okay, and the main sugars that in plants is known as sucrose. So in animals, we eat glucose, and plants actually produce sucrose, but essentially it is a sugar. Okay, so xylem is carrying water, phloem is carrying sugars. Okay, now what are the differences between the xylem? So is the xylem, is it dead or is it alive? Okay, the xylem is dead. Okay, RIP. So xylem is made up of dead cells, phloem is alive. Okay. Now, the other thing to remember is that xylem is supported. So if you imagine if it didn't have any support and water was trying to get through it, it wouldn't be able to. And the way it does that is through a special substance in the cell walls known as lignin, okay, which provides strength and support. And there are no breaks in this, okay? So it is a completely hollow tube. So you just think about your straw and how if you suck on a straw, the water comes up. It's the same thing in this xylem as well. There is nothing to stop the water from coming through. Otherwise, because of gravity, it would just fall back down. That's why the pressure of it can pull it up. Okay. In contrast, the phloem has got two things. What two cells is the phloem made up of? Which two types of cells do we have in the phloem? Fantastic. So we've got our companion cells and we have got our sieve plates. So the companion cells are just there for help, and the sieve plates are there to just allow the sugars to come through at a controlled pace and not letting everything get through in one go. Okay, guys, does anyone have any questions on that? That was basically specialized cells in a nutshell. Have you got any questions? Okay, fantastic. So, guys, we're nearly at the end of today's session. We're just going to go for a quick breather just to refresh ourselves before we move on to finally talking about cell division, and hopefully we'll have time for another quiz. So, if you guys just come back at 1:25, okay, and I will see you for our next topic. So, we see you guys back at 1:25, and we will continue from there. Hi everyone, welcome back. So it is 1:25. We have just got about 35 minutes left. So well done. I know you guys are probably had enough by now, but we've just got half an hour to go. So let's keep the momentum going. And I just want to finish off with talking about cell division, which is the last part of the foundations of cell biology. Okay, so when we talk about cell division, what do we mean? What is the point of cell division? Why do cells need to divide? Does anyone know? Fantastic. So for growth and repair, to replicate, to reproduce. Super. Really good answers, guys. So if you imagine, if you get cut by accident or you fall over whilst you're running and you start to bleed, you don't bleed to death, do you? You form a scab and new cells grow and your skin goes back to normal, right? This is because our cells are able to divide and make new cells. So our skin is continually dying and falling off, but obviously we don't run out of skin because we are continuously replacing our dead skin cells as they fall off. Okay, does anyone know the process through which we are replacing those skin cells, or how are our cells dividing? What is the term given to that? And if you have no idea, don't worry. So the way in which our cells divide, so let's say, for example, we've got one cell, okay? And the way that it becomes two cells, okay, is through a process known as mitosis. Okay, now mitosis is really actually an incredible process that is happening all of the time in our cells, and that is what we're going to talk about. So this replacement process is carried out by a cell division called mitosis. Okay, and basically what mitosis does is it produces two cells which are identical. Okay, so if you bleed from your knee and you want to make a new cell, obviously it needs to be exactly the same as the old cell, right? You don't want it to be half of that or look different. It needs to be the same. So mitosis produces cells which are identical to each other. Okay, and this process copies a diploid. So by diploid, it means that it has all of the genetic information. Okay, so it's got the information from mum and the information from dad. Now we will do a whole topic on genetics, guys, so don't worry, we will be doing that later on in the year. This is just part of cell biology. It's just the introduction of it, which comes up in your first chapter. But essentially you get two new cells which are identical to the original cell and to each other. So yeah, as we said, cell division is important for growth so that we can get taller. Otherwise, we wouldn't be able to, and also for repair. So if you damage your cells, you need to get new ones. And this is happening via mitosis, as we have already said. And you get a diploid body cell giving two new cells which are identical to each other. Okay, so basically what happens is that here we start off with one cell. Okay, now inside of our cells, we have got these bits. Does anyone know what these are? Fantastic. So this is what we call our chromosomes, okay? So your DNA, I know we've used the word DNA a lot, but DNA is actually, when we look at it closely under a microscope, it's made up of chromosomes, okay? So you get your chromosome from dad and you get your chromosome from mum, okay? Now, what happens is this is the whole process, but basically what I want to highlight to you guys is that you start off with one cell and in the end, you get two cells. Now you can see that both of these are exactly the same as the cell that they started off with, okay? So you won't end up with just like half of the DNA or like triple or anything like that. They are identical to each other, okay? They're exactly the same. Okay, so does anyone have any questions so far on what I'm talking about, or are any of these terms not making sense to anyone? Please just say if you don't understand. Okay, so let's go through this. Now, I'm getting a question which says, what is DNA? This is DNA, so this is what's sitting inside our nucleus. Okay, so let's talk about how this happens. Now, the first thing is that I think I want to actually draw it out for you guys. So I'm just going to draw it over here on my own. So if you guys grab your pen and paper, and if you have a go at drawing it too. So this is inside my nucleus, okay? And I've got chromosome from mum and chromosome from dad. Okay, now in the end, I want to end up with two cells, right, that are exactly the same as each other. Okay, so I'm trying to get from this, I'm trying to make two, okay? Let's just imagine that you're trying to get taller or you're trying to repair that wound on your knee. So you've got one cell and you're trying to get two. So what do you guys think is the first thing that's going to want to happen in order for us to get from here to here? What needs to happen to the DNA, the genetic information inside this cell? Because right now I've only got this much information, but I'm trying to get to two cells. So what can I do to try and get it into two? Obviously, I cannot just split this cell in half straight away, right? It will only give me half of the information that I need. Super. So the first thing that I need to do is I need to duplicate or double my DNA. Okay, so what I can do, if you imagine this is your cell, I will double each of the chromosome. Okay, so this will become 2, and this will become 2 over here. Okay, can you see that I've now doubled them? So I've got double the amount of genetic information. So that is my first step in mitosis. Okay, so let's have a look. So this is just another drawing of it. So what happens is that the chromosomes, they make copies of themselves and the nucleus disappears. So this is what I started off with. I've got this from mum and this from dad. Okay, and what we did is we doubled it. So now they're crossed over and there's double the amount of information. Okay, this is the first step. Does anyone have any questions on this? Okay, are you guys all following? So just say yes or no. I know this is a little bit tricky. Okay, yes, perfect. So we've doubled our DNA. Okay, now we've doubled it. So it's sort of just relaxing inside the cell and it's a bit hard to separate it, right? Like I can't just draw a line here and split them up. So what we want to do is we want to align them and just squish them all together so that it's really easy to split them up. So what happens next is that all of these will line up in the middle. Okay, so they will just line up like this. And they will be pulled apart. Okay, so let's have a look at what that looks like. So the chromosomes, they then line up. So we line them up in the center of the cell so that it becomes easy to split them up. Okay, so let's just go through that. Firstly, we duplicated them. That's the first step. Then we line them up so that they're right in the middle. Okay, we don't want them just scattered everywhere, otherwise we won't be able to separate it. We line them up and then the chromosomes are moved to opposite ends of the cell. So what happens is there are lots of forces that literally just pull them apart. So it just splits them apart into two. So half goes this way and half goes this way. Okay, so they're literally just pulled apart onto opposite ends like this. Okay, now we've split up our chromosomes. So half is here and half is over here. But actually, remember, in the end, we end up with we end up with two cells, don't we? So because we end up with two cells, what needs to happen next to go to the next step? How can we get from, we've doubled the genetic information and we've split it apart, but now we need to get 2 new cells. So how can we do that? Fantastic. So now what needs to happen is that the whole cell needs to split up, doesn't it? We need to form 2 new separate cells. So one cell needs to go over here and one cell needs to come over here. So both of them then split up. So can you see the cell is dividing? It's dividing up into 2. It realizes actually we need to make 2. We can't stay together for much longer. So they get pulled apart. And then finally, new nuclei form in each of the 2 new cells. So we wrap a nuclei around them because remember that got removed. And what you can see now is we've got 2 new cells and we started off with 1. Okay, so I'm just going to go through this whole thing again with you guys. So here we have 1 cell. Okay, this is the nucleus of the cell and inside it are chromosomes. Okay, so this is 1 chromosome and this is another one. Now we're trying to get 2 cells which are exactly the same as each other. So what is the first step that happens here? What is the first thing that we do in order to start the process? What's the first thing that needs to happen to those cells? Yay, well done guys. So you're all still awake, so they need to duplicate. Okay, we need to double them up because you can't just split them up straight away. You'll end up with half, right? It won't be enough. So we double it and we duplicate the cells so that we have double the amount of information. Then the chromosomes, they line up in the center so that we can pull them apart. So can you see they're all sitting very nicely in the middle? And then they get pulled apart to opposite ends. So can you see that they've literally been split in half and half of them are going here and half of them are going here, okay? And once they've done that, the cell then divides and we end up with 2 separate cells. And these are known as diploid because they have got the same amount of genetic information. Okay, so we've covered all of this, and this is just an overall diagram. So you guys will get a copy of these slides, and just make sure that you can familiarize yourself with them, okay? So we started off with this 1 cell over here, and we end up with 2 cells, which are exactly the same. So these are the same as this and the same as this, okay? So the way that we do that is that the chromosome makes copies of themselves, i.e. they duplicate. Okay, the copied chromosomes are then lined up at the center. They're then pulled apart. Okay, so we split them up and we push them to either side. So the original and copied chromosomes move to opposite ends of the cell. Okay, and the cell will then divide. And finally, we will have 2 new nuclei and 2 separate cells. Okay, so this is the process of mitosis. There is another type of cell division which we will talk about when we do genetics, but in the majority of our cells happening all the time is mitosis, okay? And we need this for growth and we need this for repair. So those are kind of the key take-home messages, and what I want you guys to remember is that it produces identical cells. Okay, are you guys okay on that? Do you have any questions about it? Okay, fantastic. Well done, guys. So we're just going to do a quick quiz now, as promised, just to test ourselves and to see how we have gotten on. Okay, guys, so if you all just log on, something different that you've learned in the second half of the class. Okay, we'll just wait a couple more seconds for everyone else to log on before we get started. Okay, mitosis produces what? Fantastic. So mitosis produces cells that are identical, okay? They're exactly the same as each other. Well done, root hair cell. So fantastic. So mitosis is a process where the cells create identical copies of itself. So this is not sexual reproduction and it's not to create a child. That is another type of cell division known as meiosis, but we will cover that in a different section. Okay, and you have got competition. Why does mitosis occur? So mitosis occurs whenever more cells are needed. So both of these are the same. It's a bit of a trick question. So in order to grow and to repair. Okay, well done. Mitosis purpose is to, okay, let's hope nobody gets this wrong. So mitosis purpose is all of the above, guys. So it's to produce new cells for growth and to replace the damaged ones. So all of your cells, like your skin cells or your hair cells, which have died or are damaged, we make new ones. Okay, so mitosis is doing all of these things: growth, repair, and making new cells. This is basically growing, right? These two mean the same thing, and this is repair. Fantastic. So prokaryotic cells, remember, they don't have a nucleus. Okay, so they include all bacteria and have true nuclei. Remember, guys, prokaryotic do not have nuclei. Okay. So plants are eukaryotes, guys. That is true. Okay, so for the 4 of you guys that got that one wrong, I'm a bit sad. So plants are eukaryotes, and I'll just show you guys that again here on our first slide. Okay, all animal and plant cells are eukaryotic. Okay, bacterial cells are prokaryotic. So this is true. What is xylem? Fantastic. So xylem is there to transport water and minerals. It is not there to transport food or glucose. That is the job of the phloem. What are the main materials carried by xylem? So fantastic, it's water and minerals. Okay, sugars from photosynthesis are for phloem, and sugars and minerals, these are 2 separate things. Okay, they're not carried by the same system. Okay, so the xylem of a plant transports sugars and amino acids. So guys, this is false. That is the job of the phloem. Okay, and just to say this diagram, I really like this one, and it's really helpful for your understanding. So what you guys can see here is in xylem, there's no gaps. Okay, whereas here we've got the sieve plates. In the xylem, we've got water and minerals. There are no end walls. Everything is going freely. In the xylem, everything is one way, okay? It's just going up from the root to the top of the plant, and the cells are dead. Okay, and here we've got things are going up and down. Okay, this is 2-way movement known as translocation, because the roots need sugar, but so does the flower. Okay, and so does the stem. So everything is going up and down, whether in this one it's just going up. If you have a look at the arrows, these are my sieve plates, and I've got companion cells on the sides. Okay, so this diagram is really good for comparison. Okay, what is the difference between xylem and phloem? Okay, last question, guys. So xylem transports water, phloem transports food or sugars. Fantastic. So this is the opposite, guys. This one is incorrect. I'm hoping you misread the question, because I have said this about 10 times now. Okay, so well done, guys. Fantastic. So some of you even got 10 out of 10 and 9 out of 10. That is super. I'm really impressed with that. Okay. And well done to you guys who came fourth and fifth, and to all of you really for just participating and having a go. Okay, because we're all here to learn. You don't have to get 100%. Okay, so what I want to do now. Okay, so guys, we've just come towards the end of it. So how confident do you guys feel now about this topic of cells? Fantastic. So most of you have said C and some of you have even said D. So guys, I'm really, really happy with that. I'm really impressed with everyone's participation today. Thank you all for persevering with me. I know it's been quite a long session, but basically the aims of these classes are we do cover quite a lot, okay, and we do cover a whole topic, but I don't want you guys to just leave this now. What I would suggest is the exam question packs. Make sure you do all of the questions whenever you've got the time, and we will send out the mark schemes so you can just mark it yourself, and rewatch the video. We'll send it out even closer to your mock exams. You can just go through it again. Just to revise any concepts, and yeah, and I hope to see you guys at our next session as well. Do you guys have any questions for us? Okay, I'm just going to stick around for a few more minutes for any questions, but otherwise I hope you guys have a lovely rest of the day. We'll see you guys tomorrow for our chemistry session, and please just do fill in this feedback form as well, just so we can make any changes to the class so that it's useful for you guys. But otherwise, have a lovely day and see you guys soon.